So hello friends, so let's learn some more things about the topic we are doing is proteins. Okay, so until now we have seen that on hydrolysis of proteins what you actually get on complete hydrolysis, on the complete one you ultimately get alpha amino acids. But if you don't do the complete hydrolysis, so in the mid you get peptides. So now let's learn what these are. Okay, so the head topic is peptides and their types. Okay, so let's learn something about them. So peptides are amides. The first thing is that peptides are amides that are formed by the condensation or removal of H2O. In simple words, removal of H2O of amino group of first alpha amino acid with carboxyl group of second molecule. Okay, so I'll just explain it in format like this. So you know how a alpha amino acid looks like. The alpha amino acid has a COOH group, a NH2 group, an R group and a H group. So you take one more molecule of this in the format like this: NH2, R, H, COOH. Okay, so the first thing I want to do over here is that we have to add the first molecule and this, and you have to ultimately form a compound which is a amide. Okay, so you have to remove an H2O molecule from this. So just join the amino group of the first one with the COOH group of the second one. So it means just remove a OH group from the first one and the H from the second one. So a H2O molecule is dropped out. Now form a bond between them. Do you know what is the formula of amide? The amides have a formula of CONH2 or CONH and R. So on a similar note, if you do this process, in the end you get a compound like this, and this compound has a common name of peptide. Simple. So it is just a amide compound which is formed by the addition of two molecules of alpha amino acid, and this process is done by the removal of OH from the one molecule and H from the second molecule. Now this series can be further continued. Over here I have one more amino group and one this. So if you just continue the same process, you can add one more molecule over here, one more molecule over here, then one more. So in this way you can, this compound is a dipeptide. You can also get a tripeptide, and so on, like this. Okay, and in the same way, if you just extend this process 100 times, so compounds which have more than 100 such small molecules, these are known as proteins. Okay, so now you have one term known as polypeptide. So when hundreds or thousands, when hundreds and thousands of alpha amino acids, when hundreds and thousands of alpha amino acids are joined with each other in the same format, it ultimately forms a polypeptide. An example can be shown like this: NH2 CHR CO NH CHR dash CON NH CHR double dash COOH. So this is one of the examples of a compound which comes under the section of polypeptide. So if I ask you that if the compound has more than hundreds and thousands of alpha amino acids, then it comes under this section. Okay, so you have two ends, end 1, end 2, and in the mid you have n number of similar parts. Okay, so the one end having the amino group is known as N-terminal, and the end having a COOH group is known as C-terminal, and the section between N-terminal and C-terminal, this terminal can have either same alpha amino groups or it can also be different. Okay, so suppose I have alanine, glycine, valine, lysine. So the first thing is that these alpha amino acids are shown by short alphabets like A, ALA, GLY, VAL, LYS, like this. So it might happen that in the middle you have a series like this: ALA, then GLY, then LYS, then again ALA, then VAL, and something like this. It might happen that the series is like this for this compound. Okay, there is one more term in between that is known as oligopeptide. Oligopeptides have shorter chains. So ultimately you have to form chains having n number of alpha amino acids. So you have to use alpha amino acids as small blocks. You have to attach each block with each other. You have to form a chain, and this chain, depending on the number of sections it has or the number of molecules it has, we actually show them like this. So the shorter chains, so oligo have shorter chains. They range from 2 to 10 alpha amino acids. If the polypeptides, so if you have longer chains, by the longer chains I mean more than 10 amino acids, they are known as polypeptides. And the next one, and the largest and the most important one, is the proteins. The proteins have more than hundreds of amino acids. So if you have a chain more than 100 and it has a specific structure, it comes under the section of proteins. Okay.
Thank you for watching. Visit us at needbook.in.